Good afternoon. My name is Dr. Aragon-Martin. I am the research director of Sonali Laboratory since 2012, which is the Marfan Trust Laboratory. Today, I'm going to talk about research at the Marfan Trust for the Sonali Laboratory. If you have any questions that I cannot fully address at this meeting, please do not hesitate to contact me by email, or you can Google my full name to get access to my LinkedIn or ResearchGate. Where you can find out more about Sonali Lab research, and you can also send me messages. But first, let me tell you that 3 days of the week, we are located at William Harvey Research Institute at the Charterhouse campus at Queen Mary University of London, and the Professor Panos de Lucas, where we undertake projects where families come to the Marfan clinic with clinical features like hypermobility of their joints, chronic pain, gastrointestinal symptoms, skin that is easily bruised, and hyperelastic enlargement and or dissection of the aorta in the upper thoracic region and not related to the abdominal aorta. And also 2 days of the week, we are located at the Institute of Ophthalmology, University College London, and the professor. We undertake projects where families come to the Marfan clinic with clinical features like atopic lentis, myopia, glaucoma, retinal detachment. But today I'm going to talk about the research undertaken on some of these families. We have been very busy working on 4 families that pass through the Marfan clinic. 2 of these families have clinical features related to aortic aneurysm of the upper thoracic region. These 2 families had been classified as hereditary thoracic aortic aneurysm and dissection, also known as HTAD, where Batsala, picture on the left, was able to help with this study. Batsala is an MSc student in genomics medicine from Queen Mary University of London, who also has a medical degree. And 2 other families have clinical features related to hypermobile joints. These 2 families have been classified as hypermobile Ehlers-Danios syndrome, also known as HEDS, where Sanjana and Sylvia, picture on the right, were able to help with this study. They both are also MSc students in genomics medicine from Queen Mary University of London, but Sanjana is still working on her family until December. We have screened all these 4 families with a cutting-edge technology called NGS, which stands for Next Generation Sequencing. 2 of these NGS screening techniques available are whole genome sequencing and whole exome sequencing. These 4 families were previously screened for the Marfan gene, the FEN1 gene, with a negative result. We have Roseboro family fund and BHF grant fund money to screen 2 of the HTAD families. We have some private funding to screen 1 HEDS family, and funding from Northampton University from Dr. Jamal Nasir to screen 1 HEDS family. Whole exome sequencing was used to screen HTAD family 1 and 2, and HEDS family 2, and whole genome sequencing was used to screen HEDS family 1. We cannot discuss too many details of the results from these studies because we have been accepted to present them at the American Society of Human Genetics in Washington, D.C. This November, and we have been told that it is one of their rules. Therefore, we must wait until we present our findings at the conference before we can release the information to other audiences. Here in these slides are the titles of the 3 studies we are presenting. The top left side study is going to be presented by Sylvia. Sorry, that's not right. No, the top left is actually Batsala. The top right is Sylvia, and the bottom one is me. We will be presenting Marfan Trust at this conference, where we will contact our international collaborators, create new networks, and discuss current research in Marfan syndrome and related connective tissue disorders. After the 5th of November, the results of these families will be published in either the Marfan Trust website or at my LinkedIn ResearchGate site. After processing the data in each family with bioinformatic tools, we created a list of variants which were filtered with our in-house pipeline, and the variants that were left in the list were used to screen the rest of the family members to observe if the same variant was seen in the affected and not seen in the unaffected. HSTAT family 1 on the top left side of the screen has 8 variants that are linked with HSTAT. 6 of 8 variants were confirmed not to segregate in the family, which means that these 6 variants are out of the study. And for the other 2 variants, further work needs to be done because we were not able to pick up the variant in the DNA from this family when we did Sanger sequences. HSTAT family 2 on the bottom left of the screen has 5 variants that are linked with HSTAT. 4 of to segregate in the family, and again, these were out of the study. And for the last one, further work needs to be done because although the variant co-segregates in the family, we need to explain the associated gene with our international collaborators cohort, and perhaps create family 1, top right hand side of the screen, has 13 variants that are linked with HEDS. 4 of the 13 variants were confirmed not to segregate in the family, and again, these were out of the study. 2 of the 13 variants seems to segregate, co-segregate with the family, but it also incomplete penetrance, which when family members of the family have the DNA variant, but they do not have the clinical features. And both of these variants need to be further studied. And 7 of the 13 variants, further work needs to be done because, again, we were not able to pick up the variant in DNA when we were using Sanger sequences. ADS family 2 on the bottom right-hand side of the slide has 14 variants that are linked with HEDS. Even though this family is still being under study by Sanjana, we know that at least 2 of the 14 variants do not co-segregate with the family. So that means that it's also out of the study. Also this summer, we were able to supervise a Marfan Trust student during her studentship to work in a very interesting project, which she will later explain further. But just to show here on the top right side picture, how a classical Marfan syndrome patient might look like. And if the patient is untreated, this could lead to a shorter life expectancy. While a neonatal Marfan syndrome patient who could look like the one on the bottom picture has more severe clinical features than the rest of the Marfan syndrome patients. We would like to differentiate between neonatal Marfan syndrome and infantile Marfan syndrome, which seems to be used in the literature to refer the same. This is why I would like to take this opportunity to start a discussion on the topic and propose a Marfan syndrome nomenclature to differentiate between them. NMFS for neonatal Marfan syndrome and IMFS for infantile Marfan syndrome. We know that although Marfan syndrome could be seen in younger adults, they tend to have a longer life expectancy than the neonatal syndrome patients. However, do not feel gloomy. Things are changing in the past few years with research studies and drug trials. And one of them was the 2019 Ehlers-Danios study, where we were part of that study. The following has not been confirmed yet by research studies, but it seems that neonatal Marfan syndrome patients now tend to have a longer life expectancy, thanks to the medication, and in this case, due to irbesartan. This has not been studied fully, so therefore we cannot take it to the bank yet. For my last comment, I would like to point out an update from a previous research study. The web link on the study is shown on the bottom of this slide. We published the manuscript last year, late 2022, where 2 families with thoracic aortic aneurysm and dissection were associated with the gene called LMOR1. The picture on our British family is on the left-hand side, and the Dutch family is on the right-hand side. The good news is that I have recently been informed by the Dutch group that their family pedigree is being expanded with more affected family members who are also positive for the variant that is associated with their clinical condition. This is just to show you that we are actively working also with previous studies to make sure that all those who reach Marfan Trust are getting the best outcome from our research. And just to like to say thank you to all the patients and the families involved in Sonali Lab research. Also, let's not forget the volunteers and fundraisers that always give their free time for this cause. To the Marfan Trust team, who is always helping the Sonali laboratory research studies. Students from all years, medical, PhD, MSCs, BScs, who come to our lab for training, experience, and knowledge about Marfan syndrome. And finally, the sponsors, which sometimes give that extra help to push our research a little bit further. And just open to questions if anybody wants to ask now, or if not later, fine.
May I just comment while we're waiting that most of our research projects are stimulated by questions that people have with Marfan syndrome have asked, and we take these questions home with us at night, and we lie awake thinking, I don't know the answer to that question. So we write a grant application and get some funding, and we try and answer it, and that's how we make progress. Did we have a question?
There's a lovely comment. Excellent work, Jose and team.
Thank you.
Well, perhaps we can move on then and say a huge thank you, and we'll look forward to further progress
Thank you.
in the coming 12 months.